Thank you very much. I'd like to start by conveying Karen's apologies for not being able to make the meeting. She would really like to have been able to make it, but was unfortunately had to attend a funeral. So you get to have me in her place. Whereas Karen needs no introduction, I probably need more than a little introduction. So I'm a postdoc that's been in Karen's lab for the last 5 years. And I will be setting up my own lab shortly at the University of Glasgow. And so if you like the sound of this and you're looking for a postdoc position, then come and speak to me afterwards. So I'm going to talk about the work that I've done in Karen's lab and we've done together over the last 5 years. And this work focuses on the role of P53 in cancer metabolism. So to outline my talk, I'm going to introduce the concept of cancer metabolism and how P53 plays a role in it, talk about the role of P53 in adaptation to metabolic stress, and then go on to talk about the influence of nutrients in their own right, kind of separately from P53, which is the direction this work has really led us, and then finally the implications of this work, or potential implications of this work, for cancer therapy. So to start by putting cellular metabolism in a kind of molecular biology context, this kind of cascade where we have information stored on DNA that's transferred through RNA into protein, which then dictates the function and tells you about how the cells are behaving. The phenotype is the kind of classic simplified way to look at molecular biology. And many of our schematic diagrams finish with something like this, where we have a transcription factor binds to DNA, initiates a transcriptional program that then gives us cell growth and proliferation. Simple. So really, metabolism is can be thought of as looking at this process in more detail. And actually, you know, if we don't have the metabolism underlying this process, then certainly in terms of cell growth and proliferation, not a lot can actually happen. So this is a summary, if you like, of the metabolic network, and it's pretty, and it really kind of tells us two things when we think about it. Firstly, that it's complex, but secondly, that we already know a lot about basic cell metabolism, and that's thanks to a kind of generation of biochemists who've laboriously reported on the metabolites and then the different enzymes that control these processes. So we have a good start in terms of exploring cell metabolism in the context of cancer. So when we're thinking about metabolism, we can really add a kind of layer of different biology to this kind of equation. And if the protein turns out to be a metabolic protein, we have a conversion, perhaps, of a metabolite from one to another. And then this can more closely tell us about the phenotype. And we often find that, you know, the changes that you see at a metabolite level can be so much more quicker and more accurately tell you about the phenotype than some of the more classic molecular biology. Another big advantage that we have is the analytical capacity. So we use liquid chromatography mass spectrometry to analyze metabolism on metabolites in a range of biological systems. And this is a highly developed platform, and it can quantify and identify hundreds of metabolites at the same time in a biological sample. So the analytical capacity is very good for these experiments. So cancer metabolism, the basic premise of cancer metabolism is that cancer cells have a different metabolism compared to non-cancer cells. And as we learn more about cancer metabolism, we realize that oncogenes drive specific metabolic pathways in order to have their effects. A well-known metabolic phenotype displayed by cancer cells is the Warburg effect, which is basically aerobic glycolysis, and I'll just briefly explain this. So cells take up glucose and convert it through a range of intermediates into pyruvate, and the pyruvate can either then end as lactate or be sent into the mitochondrion and fully oxidized via oxidative phosphorylation. If the oxidative phosphorylation route is used, it's very energy efficient, produces lots of ATP per glucose, but it requires oxygen. Alternatively, the pyruvate can be sent to lactate. In this case, it's less efficient, produces less ATP. However, this can be done without oxygen. So usually normal cells would revert to this type of glucose metabolism when oxygen is not present, but under normal conditions would oxidize glucose via the TCA cycle and oxidative phosphorylation. And what Otto Warburg realized a number of years ago is that cancer cells consume lots of glucose and send it to lactate even when they have lots of oxygen. So there's still controversy about exactly why cancer cells do this, but I'll go through some of the potential explanations. I should also mention that this is not just a phenotype of cancer cells, but any rapidly proliferating cells, immune cells, for example, when they have a proliferative burst, do this kind of phenotype. So one of the important things to mention is that a high flux through glycolysis could potentially promote the branching seen to produce amino acids, NADPH, and I'll talk more about this shortly. Another theory is that the lactate that's released from these cells can be used as a carbon source for other cells. Also that this phenotype can give cells resistance to hypoxia, or that it prevents OXPHOS and therefore decreases the amount of reactive oxygen species produced. So where does P53 fit into this equation? Well, P53 interacts with this small part of metabolism in a number of different ways through various transcriptional targets, and this is just an example. And overall, P53 seems to kind of dampen the flux through glycolysis, but also to support and maintain the ability of cells to do oxidative phosphorylation. So we think overall, and it's very context and cell type dependent, but overall, P53 seems to generally act to suppress the Warburg effect, which can be viewed quite basically in metabolic terms as another tumor suppressive activity of P53. So beyond the glycolysis and TCA cycle, P53 has many other roles in regulating metabolism. For example, it interacts with mTOR and autophagy. But one of the things that caught the eye of our lab was that P53 has a pro-survival role during glucose starvation. This is a paper from Craig Thompson's lab. And really because of the classic role of P53 in inducing apoptosis, cell death, this seemed very interesting to us because it was the opposite of those phenotypes, whereby under severe stress, which is glucose starvation, that cells can actually do better when they have P53. So when I joined the lab 5 years ago, my goal was to uncover other pathways and other ways in which P53 could regulate metabolism, particularly in response to metabolic stress. So the way I started these experiments was very simple. I took cells with and without P53 and exposed them to a variety of different nutrient starvations, amino acids, all sorts of different media components. What I found to my surprise was that removal of serine from the cell culture medium had an effect on both cell types, so the cells with and without P53, but the cells without P53 did a lot worse when they were starved of serine. And the reason I say this is a surprising result is that serine is a non-essential amino acid. And this means, as I already alluded to, that cells can synthesize it for themselves, and that they do this by diverting glycolytic flux. So back to the schematic of glycolysis, we see that instead of ending up in pyruvate or the TCA cycle or lactate, that glycolytic intermediates can be diverted through the serine synthesis pathway, which is 3 enzyme steps, into serine, which is then also interconverted with glycine. And taken together, serine and glycine are extremely important for anabolism. They provide precursors for protein synthesis, nucleotide synthesis, glutathione synthesis, lipids, NADPH, pretty much some of the key ingredients that any proliferating cell needs to grow. So interestingly, just about the same time I was making these observations about the desire of cancer cells for serine uptake, a couple of other labs published on gene amplifications in the serine synthesis pathway. And what these papers show is that in certain cancers, they upregulate the de novo synthesis of serine to meet their demands. But the the data that we were getting was suggesting that in some cases, basically when the cells don't amplify serine synthesis pathway, that they have a big need to take up serine from outside the cell, but also the data was suggesting that P53 has some kind of role in this process as well. So I asked the basic question, why are the P53 null cells sensitive to serine starvation? So I'm going to briefly explain one of the most important techniques that we use in conjunction with mass spectrometry, which is using labeled carbon, so carbon-13, and tracer studies. So natural glucose and most carbon that occurs in nature is carbon-12, but about 1% of natural carbon is a slightly heavier isotope, not radioactive, just heavier, which is carbon-13. And normally it's, as I said, only present at about 1%, but you can buy glucose or various other metabolites where the carbon-12s have been swapped for carbon-13s. And this is useful because with mass spectrometry, you can see a mass shift so that you can trace where this glucose goes. And we basically say that when we do the analysis, that the unlabeled glucose or unlabeled metabolites have a +0 mass, so they're the natural mass. But then things that come from the labeled metabolite then have a shift. So fully labeled glucose would be a +6. And then this allows us to put a time component to our analysis and see how quickly does this labeled glucose feed into various different pathways. So this is the serine synthesis pathway. And so what we would see, if we look at these various different metabolites, for example, in fructose phosphate, we would see how quickly does the N+6 version of fructose accumulate from this labeled glucose. We can also then look at lactate, which would be just an N+3 because it's only 3 carbons, and serine, for example, So we can look at the rate of serine synthesis by using this method. And interestingly, we find that in the P53 wild-type or null cells, they both do serine synthesis. And when we look at the expression of the enzymes in this pathway in RNA and protein level, they both get upregulated in the P53 competent and in the P53 null cells. So there didn't seem to be a problem with the ability of these cells to de novo synthesize serine. What we did see, however, and some of our work is in collaboration with A.L. Gottlieb's lab on this, who introduced this kind of analytical technique to the Beatson. And what we found was that there's a blockage lower down when we starve the cells of serine, and this is at the level of PKM2. And that has quite important implications, and I'll come on to those. So we could see that the P53 wild-type and P53 null cells could make serine, both make serine. So then we looked further downstream to see whether maybe these cells had changes in their ability to handle the serine that they had then made. And this is where we kind of made a breakthrough. And what we discovered was that after serine is converted to glycine, 2 of the most important pathways after that are the synthesis of glutathione, which is an antioxidant, protects the cells from reactive oxygen species, but also then as glycine is fed into nucleotide synthesis or purine synthesis via IMP. And what we see very clearly is in the P53 wild-type cells starved of serine and glycine, they maintain their glutathione synthesis, but they stop their nucleotide synthesis. Whereas the P53 null cells attempt to do both processes at the same time. And by doing both things at the same time with depleted serine, they don't have enough to do either properly. And over time, the consequence of this is that the P53 wild-type cells build up. So they have lower glutathione immediately after serine starvation, but then over time they build their glutathione levels back up, whereas the P53 null cells are not able to do this, and they suffer from reactive oxygen species damage. So in this case, P53 helps to control the balance between nucleotide and glutathione synthesis, and P53 has well-characterized functions in terms of inhibiting nucleotide synthesis through its cell cycle control, P21, etc. So to summarize, all of the data we've generated around this adaptation to serine starvation, if we start with the idea that some cancers have enhanced de novo serine synthesis, but the other cancers don't have this, and then become dependent on the uptake of exogenous serine, and that this is converted into glycine, which amongst other things is important for nucleotide and glutathione synthesis. Now, this ability to take up serine from outside the cell makes the cells more efficient because they don't have to divert their glycolysis anymore. However, it makes them then dependent on exogenous serine. so that when we take serine away, the cells go through a metabolic stress. Normally, serine maintains PKM2 activity, and that when we starve the cells of serine, this decreases PKM2. This then decreases the flux to lactate, which allows the cells to increase the amount of glycolytic intermediates fluxed into the serine synthesis pathway. This then causes an energetic stress, meaning because the cells are now making less ATP, they have to use the pyruvate more efficiently, which means they divert it into the TCA cycle. But the unfortunate consequence of this, through more oxidative phosphorylation, is the generation of more reactive oxygen species. So where P53 has a role via P21 is to block nucleotide synthesis, facilitate the continued glutathione synthesis, which then rescues the cells and prevents the cells suffering the stress of the reactive oxygen species. So in summary, in this context, so serine starvation alters glycolytic flux and induces reactive oxygen species, and P53 promotes adaptation by controlling the cell cycle and nucleotide synthesis, channeling the limited serine into glutathione. And we can, based on this, taking the P53 null cells, which again don't grow very well at all without serine and glycine, if we add pyruvate to help them overcome this glycolytic diversion, but then also add additional glutathione, which protects them from the reactive oxygen species, they can now grow pretty well without serine and glycine. So that's wild-type P53, but as we've already heard, many cancers express a mutated form of P53. So this is some new data we have on the role of mutant P53 during serine starvation. So we've already seen in wild-type P53 cells, they do okay when serine is removed. The P53 null cells don't do very well at all. But interestingly, in isogenic cell lines, and these are Burt-Vogelstein's cell lines, isogenic cell lines that express mutant P53, either with wild-type or on its own, that they do even better when they're starved of serine and glycine. So we were very interested to try and work out what's going on here, and this has been vexing us for a while. And basically, if we look at the model of how wild-type P53 works, it's via P21, and we're fairly certain that mutant P53 cannot act via P21, and many studies have shown that the mutant P53 doesn't activate P21, and that seems to be the case in our cells, certainly. So we're wondering how mutant P53 can phenocopy the wild-type P53. So really interestingly for us, we see at the metabolite level, and again using labeled glucose, we see that the wild-type P53 and the mutant P53 cells behave exactly the same at a metabolite level. So they sacrifice nucleotide synthesis, so a decrease in nucleotide synthesis, so that they can maintain glutathione synthesis. And whereas in the P53 null cells, they really don't recover their glutathione, the mutant P53 cells are able to do this. So we kind of scratched our heads and we're wondering how this could happen. So we decided to try and conduct an siRNA screen where we would starve the cells of serine and compare wild-type P53 and mutant P53 expressing cells and look for an siRNA that would perhaps specifically inhibit the growth of the mutant P53 cells compared to the wild-type P53 cells, and hope to find a target that maybe was working in cooperation with mutant P53 to allow the mutant P53 cells to survive during serine starvation. So for the screen, we wanted to have a positive control that could differentiate between the mutant cells and the wild-type cells. We thought nutlin would be the obvious choice. And so you can see here that during serine starvation, or not during serine starvation, you treat the wild-type P53 cells with nutlin, and they increase their levels of wild-type P53, cell cycle arrest, and you have much less cells. But in the mutant P53 cells, the nutlin does stabilize the mutant P53, but it doesn't act on P21, and it doesn't initiate the same type of cell cycle arrest, and the cells consequently do just fine. And the siRNA screening facility guy said to us, well, that's great, but we really want an siRNA that's a positive control for our experiments. So we thought the obvious choice then would be to mimic nutlin, which would be to knock down MDM2, and that would be a nice control. But best laid plans, and that didn't work at all. So we saw that when we inhibited nutlin in the wild-type P53 cells, we got what we were hoping for, which was a decrease in proliferation. But also then in the, surprisingly, in the mutant P53 cells, knocking down MDM2 really caused the mutant P53 cells problems. So this is very much a work in progress, but we think that MDM2 seems to play a pro-survival role in the mutant P53 cells. And I think Lauren will also talk about this, the potential role of MDM2 in metabolism. And like I said, it's still a work in progress, but it's, I think, a very interesting observation. So in the course of these experiments, a lot of work on serine, but also a little bit about glycine, and made some quite interesting observations independently of the genotype of the cells, independently of P53. And so they're both non-essential amino acids. As I've said, they can both be made from glucose. Serine has 3 carbons and glycine just 2, and they can be interconverted by the addition or loss of this carbon. And this turns out to be very important biologically. So what I had found was that in what is essentially complete media, which contains serine and glycine, the cells grow very well. And if you take serine and glycine away, so that's the gray line here, various cells do less well when you take away serine and glycine. But if you add back various different amounts of serine alone, the cells fully recover their growth. However, if you add back just glycine alone, you have the opposite effect, and that the cell proliferation actually diminishes the more glycine that you add. This is a very striking, clear phenotype. So, as I said, we saw that glycine could not compensate for serine, and that glycine without serine has a growth inhibitory effect. So I looked in the literature and obviously looked to see if anyone had published on this before, and found some papers from 99 which fed mice and which had various tumor models, excessive amounts of glycine, and showed that just the dietary glycine could inhibit tumor growth. So I thought, okay, well, that sounds fair enough based on what I've seen. But then another paper caught my eye from 2012, which was a science paper showing that glycine uptake was correlated with rapid proliferation. And another showing that glycine cleavage, again 2012 in Cell, showing that glycine cleavage supported tumor initiation. So to me it seemed surprising based on my observations and these observations that glycine uptake could promote proliferation. So I wanted to explore this relationship further. So in thinking about the interconversion of serine and glycine, when serine is converted to glycine, you also produce this one carbon, which I just mentioned. And this is a very important metabolic precursor. You can also obtain it by cleaving glycine itself, which is by the glycine cleavage system. And the one carbon then feeds into the THF cycle, which is very important for nucleotide synthesis, methylation reactions, and a host of other things. So in theory, you can get these one carbons from glycine or from serine. So they're, in theory, metabolically interchangeable. So what I started by doing was looking at the expression of these enzymes and whether they changed or not during the different nutrient levels. So whether you have serine and glycine together, no serine, no glycine, high serine or high glycine, you see absolutely no difference in the enzyme expression at all. And this kind of harks back to the introduction where I said, you know, protein levels are one thing, but the metabolites can be doing completely different things. So what we're looking at here is something that's really independent of the protein expression levels. So then we move to our favorite technique of using a labeled metabolite to trace the metabolism of glycine in these cells. So here on the left, the cells are fed serine and glycine together, or glycine only. And we see that when we feed the cells, they take up the glycine. And just in terms of the glycine levels, it's quite similar between the different conditions. These cells maintain their ability to synthesize glutathione. However, their ability to synthesize nucleotides is vastly diminished. I thought this could be explained really if we were seeing an increase in the conversion of glycine to serine. Because if we had more synthesis of serine from glycine, it would consume one carbon units and prevent nucleotide synthesis. But here you can see it appears that very little of the glycine is being converted into serine during these conditions. So I kind of didn't believe that result or technique was quite telling us the truth. So what I had to do was develop a technique, we called it pulse stop flux, because basically during the labeling experiments, I would then add a very short flux of unlabeled serine into the cells to saturate the serine levels, which would allow the accumulation of the labeled serine, which I was sure was happening. And what we actually see when we do this technique is that instead of seeing nothing, we do see the accumulation of serine that's being synthesized from glycine. And the more glycine we have in the medium, the more serine is being made. So this reverse reaction does indeed happen. So the model that we have here is that when cells are fed serine only, they convert it into glycine and 1-carbon units, which promotes nucleotide synthesis. Whereas when the cells are fed glycine only, they have plenty of glycine, but the 1-carbon units get sucked in to produce serine because of the equilibrium of the reaction changes. And that this effectively has an antifolate activity. So excess glycine has an antifolate activity. And then if we contrast this to the previous papers that I referenced, actually what happens is that glycine uptake is a consequence of rapid proliferation rather than a driver. And so the cells that most rapidly proliferate consume all their serine in a cell culture experiment first, then switch to glycine. So it seems that glycine uptake is promoting proliferation, but really these cells use the serine first and then move on to the glycine because they proliferate so rapidly. And actually glycine cleavage in the cells that we look at, rather than providing 1-carbon units for proliferation, for nucleotide synthesis, glycine cleavage is useful to detoxify glycine, to prevent the buildup and the inhibition of proliferation that we see. So that's all very interesting, but is it of any use? And is there any way that we can take these observations and apply them to cancer therapy? So in our initial paper, we had done an experiment where we'd aimed to really replicate what we'd done in the cell culture dish in mice. And we did this by injecting the same cells, the HCT116 cells, into mice and to form xenografts. And of course, I naively hoped that if I could just treat the mice like a cell culture dish and take away the serine and glycine from the diet, that this would translate into a phenotype. And surprisingly, it did. And so that when the cells are fed a complete diet with serine and glycine, the P53 wild type and null cells form tumors equally well. But then when we just change the diet and take serine and glycine out of the diet, we inhibit tumor growth, and this is more significant in the P53 null cells. So we were quite surprised that this actually worked. And also then, because of this change in tumor growth, we were able to see an increase, a significant increase in the survival of the mice on the serine and glycine-free diet. So xenografts are very well, but they're not the best model for tumor growth in humans. And what we have at the Beaton is a number of genetically engineered mouse models that more closely recapitulate human cancer, and tumor development in these models is spontaneous. And I know Arnie's going to talk more about the lymphoma model here, but these mice basically express MYC oncogene, which drives the spontaneous formation of lymphoma. And in these mice, what we decided to do, instead of changing the diet early on, we thought we would allow them to essentially develop small tumors and leave it as late as we could before we changed the diet. So you can see at 60 days, just after 60 days, the mice start to come down with tumors. So that's when we change the diet. And just by changing diet, and the caloric levels are the same between these two diets, so it's not a caloric restriction, it's just a change in the amino acid composition. And the total protein levels are the same, the total amino acid levels are the same. And what we see is that just by changing the diet, removing serine and glycine, we can significantly improve the survival of these mice. Doesn't always work. So this is a pancreatic model that we've already heard a little bit about. And interestingly, in this model, when we change the diet again at 60 days, we don't improve the survival of these animals. And there's been some recent work from Daphne Barsagi's lab showing that KRAS-driven macropinocytosis feeds PDAC tumor growth. And macropinocytosis is the process whereby cells can take up whole proteins and chop them up and derive their amino acids from whole proteins instead of taking free amino acids from the serum. So we think that there's a good chance that in the pancreatic cancer at least, they don't worry so much about the free serine levels in the blood. They're more concerned with taking serine from proteins, and that's maybe why that they're not susceptible to our diet. In an intestinal adenoma model, which is the APC-min model, We also see that diet, and again here we're changing diet at 80 days, so that these mice already have tumors, that just changing the diet can significantly improve survival. So we don't think we're going to start taking patients off chemotherapies and saying all you need is a diet. I think anyone would be sensible not to go down that route. But what we hope to do is to be able to think of ways that we can use the diet to enhance the chemotherapeutic activity of other drugs, whether they are traditional chemotherapies or emerging novel chemotherapies. So an example of how we hope to do this comes from the treatment of the cells with oligomycin. So I already said at the start that in adapting to serine starvation, the cells are forced to do more oxidative phosphorylation. So if you can inhibit oxidative phosphorylation at the same time as starving these cells of serine and glycine, you can actually enhance the inhibitory effects on cell proliferation. So that using a low dose of oligomycin, which inhibits mitochondrial ATPase, you see that very low dose doesn't really affect the cells if they're grown in complete medium. But if you drop serine and glycine out of the medium and treat with a low-dose oligomycin, you can really strongly inhibit the growth, and these are the P53 wild-type cells. And oligomycin is not really a drug you could, or that's used in people, so another way of inhibiting OXPHOS is with metformin. So metformin is an anti-diabetic drug that's receiving more attention now as a potential anti-cancer agent, and it also, as I said, inhibits OXPHOS. And we find a very similar result, so that when we combine serine and glycine starvation with metformin treatment, we can have this synergistic or additive effect in terms of inhibiting cell growth. So the proof of the pudding, again, is whether this can translate into a genetically engineered mouse model that spontaneously generates tumors. So we've already seen in the lymphoma model, in the broken line here, that on a controlled diet, the mice die. Most of them are dead by 300 days. Now, if we treat these mice with fenformin, which is a similar drug to metformin, has better bioavailability, fenformin on its own doesn't really have much of an effect in terms of the survival of these mice. However, if we look at the broken line again, this is the effect that we've already seen of diet alone, so that we know that the diet improves the survival of the mice. But then when we combine the fenformin along with the diet together in these mice, we see a significant improvement in survival. So, of course, mice are mice, they're not humans, so there's still a long way to go in terms of working out whether we can translate these dietary studies into potential chemotherapy or ways of enhancing chemotherapy. But we're quite pleased with the data we've got from the mouse models in suggesting that this might be a possible thing to do. And other labs have started to use the diet as well. And so in Michael Pollock's lab, they've also shown in a mouse allograft model that serine starvation diet enhances the effect of fenformin also. So this has been replicated. So to finally summarize what I've talked about, we've seen that P53 has an important role in response to metabolic stress. And in contrast to its kind of traditional apoptosis or senescence kind of role, it can often promote cell survival. And we think this is a lot to do with the fact that in metabolic stress, often the genome is not threatened as much as in a genotoxic damaging stress scenario. Our preliminary data suggests that mutant P53 can phenocopy wild-type P53 in the metabolic context of serine starvation. We think MDM2 has a role, but we're still working on this. If we just look at serine and glycine, they're theoretically metabolically interchangeable. However, the exogenous levels of these separate amino acids can have dramatically different effects on proliferation. And finally, we've shown that the dietary intervention, just by manipulating amino acid levels, has therapeutic potential in cancer that we're very interested in and continuing to work on. So just to acknowledge, obviously, Karen, it's been a real pleasure to work in her lab for the last 5 years. And the other members of the lab who've really contributed to this work, particularly are Christian and Celia. A.L. Gottlieb's lab, who work closely with ours, has helped us a lot through these various different experiments. Karen Blythe, a lot with the in vivo work. And then our metabolomics co-facility with Gillian and Niels have contributed a lot to the metabolomics analysis. And then funding has come from CRUK and ERC. And thank you for your attention also.